And I will bow out.
Hi, everyone, and welcome to our Fall 2024, Winter 2025 webinar series. My name is Elizabeth Grasso Kelly, and I am the current president of Phi Tau Sigma and the past chair of the program committee. Today, we are pleased to offer this webinar to our members and guests. So this fall, we had a variety of webinars that we've been presenting on a monthly basis, starting with Dr. Ale, who did a novel 3D food printing approach to generate micronutrient delivery systems. In October, we had Dr. Tim Duncan and Dr. Scott Coffin with microplastics and nanoplastics. This month, we are presenting with Dr. Joe Regenstein, writing with intent to submit a peer-reviewed paper. And next month, we have an offering from Dr. Matthew Moore, Dr. Ogachukwu Taisai, Christina Allingham, and Dr. Claire Zollner on abstract preparation and best practices for presentations. I'll be sharing more about that webinar at the end of today. Looking forward to today, I'm happy to have everyone here, either online or watching this at a later point. Today's webinar is open to all, and we'll be providing the recording via our YouTube page and later on our Phi Tau Sigma website. Today's webinar is really based towards our student member audience to help develop real-world professional skills. Today's live webinar will cover suggestions and best practices related to writing and submitting a peer-reviewed paper. Specific takeaways for the audience include understanding impact factors, preparing a manuscript and abstract, determining authorship, and considerations for citations and references. I'd like to give a special thanks to the Student Relations Committee for organizing this event. Our speaker today is Dr. Joe Regenstein. He is a professor emeritus of food science and head of the Cornell University Kosher and Halal Food Initiative within the College of Agricultural and Life Sciences. He's also an adjunct professor in population medicine, in veterinary medicine, and Jewish studies, arts and science. He's an adjunct professor of food science at Kansas State University. His BA and MS are from Cornell, and his PhD in biophysics is from Brandeis University. Dr. Regenstein has received IFTs, Elizabeth Steyer Humanitarian, Borlaug International, and the Dr. Carl R. Feller's Career Service Awards. He co-founded and edited IFT's Religious and Ethnic Foods Division newsletter and wrote and edited the aquatic newsletter, Foods newsletter, for many years. He's an IFT fellow who served on IFT's executive committee and was IFT's first congressional science fellow. He is a lifetime member of Phi Tau Sigma and a guest fellow of New Zealand's IFST. Dr. Regenstein is co-editor emeritus of Food Bioscience and founding co-editor of this publication. This is the first English peer-reviewed Chinese food science journal. So at this time, I'd like to hand it off to Joe and allow him to share his screen as soon as I stop sharing.
Thank you, Elizabeth.
The floor is yours.
Yep. Okay, there we go. Thank you all for attending and or listening in after the fact. It is my pleasure to go over this material. It came, as Elizabeth said, from editing Food Bioscience. I took a leaf from comparative reviews in food science and food safety. And basically, as the editor, I read every accepted paper and did that for about 8 years as we grew the journal. That means I got to see papers that were accepted and still often required multiple rounds of editing. So I'm a very critical editor, and I tried very hard while editor of Food Bioscience to maintain what I considered a fairly high standard. Did frustrate some of the authors who knew the paper had been accepted, but were still being asked to make improvements so that it was an appropriate, proper material. I obviously come with the bias that comes with food bioscience, and many of the things I will say are obviously useful for any peer-reviewed manuscript in any field. Some are very specific to food science, and where I've taken examples, they may be relevant to food bioscience. So I've tried to keep it more general. But the most important thing from your perspective is the first thing you should be doing is following the instructions of the journal that you chose. And we'll talk about that as we work through this. But again, what is a peer-reviewed paper? I like to think of it as a story. Do you have something to tell the rest of the world that is a worthy piece of research, and have you learned something useful? And collecting data per se is not what science is all about. It at times is extremely useful and extremely important, and there are often mechanisms for publishing data sets that were collected, summaries of data from hospitals or quality control labs, and they are very useful and important. I'm not putting them down, but we're talking about original research with a story that pulls together the material and ideally avoids what is often referred to as the least publishable unit, which is a case I saw at one point early in my career where one of the students in a related field had looked at 8 different pesticides. And instead of writing one paper summarizing the whole thing, which would make a strong story, he chose to write 8 separate papers to get 8 papers on his resume. And I believe that that actually worked against him career-wise because he clearly was repeating a great deal of information because they were all done the same way, just with each of the different pesticides. So that is something to think about in terms of the impression you're going to make on others. Remember, you're telling the story to get the information to others. And a peer-reviewed paper is a highly structured format, and it is not the natural way you would tell a story. We all learned as children to say in the beginning, and then we start, and they lived happily ever after is how we finish. And that just doesn't work for a scientific paper. So it's a very stilted way of presenting it, but it structures things so that you don't have to create a format for telling your story. So the first thing I said was to follow the instructions of the journal. Well, how do you get the right journal? It should be a journal, ideally, that you're reading and using, and that a number of the papers directly related to your field will be citing those articles from those journals. That is, the journals that, like Journal of Food Science, which is our IFT journal, which is specifically food science and is an important source of material. And so you want to also sometimes fine tune it to a micro or food chemistry or food micro or food engineering so that, again, it will reach the audience you want to reach. And as mentioned in the introduction, this question of impact factor. The impact factor is something about the journal. It is how many times the average paper in the journal is cited. Long term, you're much more interested professionally in the citations to your own paper. Now, a higher impact factor means there's a likelihood of more people reading it, but if the wrong people are reading it, they're not going to cite it. So you want to have the right paper. Now, if you've had something very, very exciting, you might want to go for one of the science journals or one of the nature journals that have this very wide audience, very high impact. But it doesn't mean that your paper is going to be cited just because it's in science or nature. And the peer review process with those journals is extremely tough, and the number of papers accepted is extremely low. So again, particularly those of you who are still students, you want to get your paper published, and you want it timely at times. And depending on the rules in your university or your professor, you sometimes have to have a paper. But the goal is to get the paper accepted and printed and cited. And remember, half of the papers published in the world are never cited, which is a reminder that there are a lot of weak papers out there. You don't want yours to be one of them. So think long and hard about the right place, the people who you're citing, what journals are they going to, and what are the people in your lab reading and talking about and using for journal clubs or presentations. Those are the journals that represent the aspect of your work that makes it likely you will get to your readers. And the order of a scientific paper is structured, again, as I said. So I want to go through these structures and give you some hints and things to consider. The title page has obviously the title, the names of the authors, the location of the authors, and the format of that should be appropriate for that journal, and it has the corresponding author. Now, the corresponding author is one thing in reality from what it was and is meant to be. The corresponding author, I have always considered a punishment. It is the person who has to actually do the work of submitting the article, of being in touch with the editors, of submitting the revisions, writing a letter to the editor if they need a time extension, all of what is the mechanics of submitting a paper. It is not an honor. It is a job responsibility. However, I have learned that in certain countries, that corresponding author is a must for the principal in the laboratory, so that the corresponding author is often not the person actually doing the work. And I have come to the point of view at this point that I accept the fact that it has to be the senior author in the laboratory, to the point that I would argue that person doesn't have to be an author on the paper, because sometimes it's inappropriate for the senior person who may or may not have had anything to do with the paper to be an author. The question of authors is a topic that needs to be thought about. It needs to be people who've actually contributed, and it needs to be people who are willing and able to understand and take responsibility for the entire paper. So somebody who gives you an electron micrograph on their very expensive equipment deserves an acknowledgement. They are not an author unless they are ready to take responsibility for the entire paper and really have read and understand the entire paper. The NSF, National Science Foundation, has an entire book dealing with some of these very itchy issues, with a long section on qualifying who is an author, and even more complicated, deciding the order of the paper's authors. And that, again, is a touchy issue in many places. Generally, the person who did the work, really did the work and wrote the paper, is the first author. The senior author, which is, if the professor has been involved, that is normally the last person on the list. That's always been my rule. When a student didn't write the paper and I had to write the paper, I then sometimes made myself first author because I was writing and interpreting and doing the guts of what is a scientific paper, but the student had done the research and written the thesis and therefore was often then, in that case, the final author. So that is one page with that information. Think about your title carefully. I try to keep it as short as possible and don't use, and I'll say that a number of times in this talk, don't use extra words to fluff it up. A study of. All that does is make it harder when people are scanning lists of paper, a study of, a study of, a study of, research on. Get some keywords right at the beginning of that title so you catch people's eye for the people you want to catch. The abstract is the most important information about the paper. It basically takes the paper and hits what was done, why it was done, and what was learned. And it should be upon length for the journal, and the shorter the abstract, the harder to write. Most of us can write loosely. It's filling a specific number of words that is much harder. It is, unlike an abstract for a meeting, it is best to write it after the paper so that you really are consistent with the paper. You should avoid undefined abbreviations, and we'll talk about abbreviations a bit later. It is a standalone. I learned early in my career that certain trade associations, for example, will circulate abstracts to its members, totally independent of the paper. So again, you want to use the abstract as a marketing piece, in a sense. The person reads the abstract, you want that abstract to say to the right people, read my paper. This has whetted your appetite, now read my paper. And you want real information, not promises. And that also goes for review papers. Don't promise. We will be talking about, we will review the importance of apples. That's not useful to somebody reading the abstract. And in some cases, in other countries, less developed countries, people may not be able to get the whole paper and may want to cite the abstract. So again, there's a proper way to cite an abstract and by the author, but it also means that you get the information to places where they may only have access to the abstracts. Keywords used to be more important. With modern full computer searches, they're less important, but they still need to highlight what are really the important things for your colleagues. They're not methods. Nobody cares as a keyword that you used a spectroscope or you did protein determinations or you measured structure. What you want are the materials or what else is unique, which enzymes you're studying and where they came from. And often you want to put in the Latin name. And the Latin name should probably appear in the paper once, but it is a good search term because that's the universal thing we've all agreed that whatever animal or plant we're talking about, its Latin name is what is common for everybody. The introduction should focus on information that is needed to understand the current work. It is not the literature review from your thesis. That, if it's really a good literature review and is something that hasn't been done recently by anyone, it is a review paper. They are wonderful. They are even more heavily cited, so that's good for your career. But that is not what goes in the peer review paper. It should focus on what is needed to understand the current work and why are you working with fish or tilapia, I'm a fish guy, or sea bass or catfish. Why is that important to the reader. And then at the end, the last paragraph is your objectives. And if the literature review is well written, you should be able to predict what the objectives are, because the introduction should set up the research perfectly, and therefore the objectives should be unbelievably obvious. And again, the other question you want to ask again is, why should I, the reader, care? Why would anybody want to pick up this paper? So again, they've gotten past the abstract, they've read your introduction. Are they going to stop there, or are they actually going to read the paper? And don't pad citations. There's a temptation to cite your own work, to cite your laboratory's work, in some cases to cite the school or even within a small country. The citations should be exactly throughout the paper what is needed to properly write the paper. It is not about adding citations to help your friends improve their citation index and improve the journal's impact factor, etc. I am aware in some foreign countries where a journal is supported by an organization, that members of that organization are actually rewarded if they help increase the impact factor of a journal, which again is why these things should not over-dominate. So, and just one thing, I'm very sensitive. You'll see there are certain types of words I get hung up about. Nothing is obvious. Remember that your reader may be a senior scientist who has worked in the field for 30 or 40 years, but it also may be a graduate student or even an undergraduate student where your paper is the first paper they have ever read. Scientific paper, obviously, peer-reviewed paper. Think about that. It can be that entire range of people. So don't say it's obvious, because an undergraduate reading his or her first paper is not going to necessarily feel it is obvious. Avoid that. If it needs to be said, justify your statements. And again, the same point that I just made, I want to highlight in terms of the writing overall. Keep your language simple. Avoid emotional words. And I'm a real bug about that. The data doesn't reveal something. The data shows something. And don't be trying to come up with synonyms for these key words like show, the results. Keep it the same word, because a person whose language is not English, keeping the same words makes it easier to read. If you keep changing the words, people are saying to themselves, well, is there a difference between when the data reveals something versus the data shows something? Is the author trying to say something important by his or her choice of words? So again, be consistent and think about your writing, and is it sloppy? Because when I see sloppy writing, I say to myself, was the lab work sloppy? And yes, sloppy is an emotional word. I'm reacting emotionally to the fact that this person has not done a good job with the presentation. It makes me worry about the data itself. Use the spelling and grammar checkers. The new ones are even AI based on some of the word, but don't count on them. There's a skill called proofreading. That means after everything is done, you reread the paper a day or two after you wrote it to catch mistakes. Ideally, you'll have a colleague ideally in the lab, though even it can also be a spouse at times, or a girlfriend, boyfriend, or whatever, who you can ask to read it to see if there are any mistakes. And be careful as you correct mistakes not to make new mistakes, because one of the challenges there, especially with abbreviations and symbols, is to remember which one you're using in your paper and making, if you're new at this, making a list of abbreviations and symbols you've used so you're consistent is helpful. The methods and materials. Here's where I'm an analytical chemist at heart. I consider that the heart of the paper. If I don't know what your methods and materials are, or I have questions about them, I have no faith in the data. So the better you handle the methods and materials, the more faith the reader has in the results and discussion. Now, I know there are some journals that de-emphasize the methods and stick it at the end. I think that's a mistake. I think that science is about the methods and materials to understand the universe, to understand nature. And again, a criteria I use is, can a graduate student or undergraduate at the other end of the earth duplicate the work? If you're doing the work in Argentina, can somebody in China duplicate it? If you're doing the work in Thailand, can somebody in also in Argentina or Brazil or Peru duplicate the work? And that's where it gets tricky in food science. What are your starting materials? Are they sufficiently characterized so somebody knows what you are using? What is an orange? What is a tilapia? An orange is a fruit on a tree. There are many breeds. Every year, if the same tree is used, those oranges will be different. If you harvest it today, tomorrow, and the next day, those oranges will be different. So the more you can tell people, the more they will understand your materials. Did you go to a supermarket? And sometimes that's perfectly appropriate. I'm not putting it down, but you need to admit you went to a supermarket and you bought something where you don't know its history. When you're doing your results and discussion, think about that. Did you sample something like tilapia, even if you're, let's say, working with a particular aquaculture farm where you could get fish? Did you do it one time at one time of the year, or did you sample 4 times a year to cover the seasons? If it's an outdoor facility, did you sample even more often? So again, is buying one bag of oranges, even if you did 3 repeats of, you took one orange for each of your tests, different oranges? Or did you take a bunch of oranges and put them in a blender and have a composite sample? All of those things are extremely important in understanding how valuable your results are. None of us have ever studied universally oranges or universally tilapia. It is a very limited study. Even if you took samples from 10 farms 10 times a year, that's still not the world of the whole world of that material. So be a little more modest. One of the things I talk about is scientific modesty. Science is a collective effort. You're collectively adding to the information, but don't over aggrandize yourself. You've done a nice piece of work. Let the rest of the world see it and let them decide. You don't have to tell people how wonderful you are and how wonderful the research is. Give the methods sufficient that people understand what you did, even that undergraduate reading his or her first paper. If it is standard method like AOEC, AOCC, or BAM, you can at least tell which method you used, and those have numbers. There are more than one way to do a Keldol, for example, so give a full and proper reference. And the same has to do with materials and equipment, and software is a piece of equipment. You need to think about the standards of your chemical. You need to understand that the supplier of the equipment makes a difference. And again, the tradition is we put down the location of the supplier or the software company, etc., and give its location once per company. For those of you who are listening internationally or are from other countries, in the United States, for sure, we have the same name for the same city in different states within our country. So, for example, I grew up in Newark, New Jersey. Some of you go to the University of Delaware in Newark, Delaware. And up here in upstate New York near Cornell, we have a Newark, New York. So just on the east coast of the United States, we have 3 Newarks. So you need to put the state. And the purity of some chemicals does make a difference. For some purposes, urea and ammonium sulfate, for example, will work differently if it's reagent grade or if you use a higher grade. So even the standard reagent analytical grade is sometimes insufficient for getting reproducible, consistent results. So you need to indicate not only what chemical, but what chemical standard it is meeting. And again, if you've modified the original method or you've used a specific piece of instrumentation when there are choices, you need to let us know that. Time and temperature. Room temperature fluctuates. 25 degrees, which a lot of people report, plus or minus 2 degrees. 77 degrees Fahrenheit is a darn warm room. We keep our air conditioning in the summer at that temperature, but most of the year our house is more like 20 degrees, and most labs are not 25 degrees. So you should keep track of the temperature a little bit. How long did you store the intermediate materials? You freeze-dried something and put it in the freezer. Was it there for a week, a month, or 10 years? That is important. A very specific problem, centrifugation, you need to describe it in G-force. That's the ruling. It's the bottom of the tube. It is a questionable number. You can put RPMs once in parentheses, but it is important to indicate which centrifuge and which, ideally, which centrifuge head, because that affects G-force. Obviously, your blanks and controls, particularly positive and negative controls, and in many cases, issues of recovery of any material ought to be done. And one of the areas where I drive people crazy, and unfortunately, I feel that in food science, it is well accepted to do amino acid analysis and fatty acid analysis poorly, because what they're doing often is looking at the total peak area as given to us by some computer software. And then each of the peaks is identified and is a percentage of the total peaks, which means that when you're finished, it adds up to 100%. When you're forced to do it quantitatively in terms of milligrams of amino acids and have standards for each amino acid, many times people discover that their hydrolysis procedure recovers 50 to 60% of the so-called protein content. And hydrolysis of amino acids is not just loss of tryptophan. A number of amino acids or pairs of amino acids, peptide bonds, do change over 24 hours and are affected, though not completely disappearing like the tryptophan. Are you getting your information from the manufacturer? Say so. Are you using computer software? Supplied with the instrument is fine. But how was the area under the peaks measured? You don't know most cases. So again, be a little cautious and say, as supplied by the instrument, because there are multiple ways to measure the area. All the different programs are not doing the same thing. And again, have you run the proper control? Are you using the right statistics? Consult a statistician. Identify the sub-programs you're using. Again, attention to detail. A few other comments. SDS gels, Kamasi Blue, saturates at an unbelievably low level. Most publishable SDS gels are saturated. So if you're running quantitatively, whether you're doing it directly or via a photographic program, you are way outside the linear range. It is not quantitative. Many measurements are equivalent. So you're doing a calibration curve and it's equivalent to so much of something. Think about what that means. You could have a very different amount if one of your compounds reacts differently to the test process. And that's an example is protein. It's either crude protein with some magic conversion factor, or you're comparing it to BSA. So it's really a BSA equivalent. I would argue that none of us in routine testing for protein, whether it's Biuret or Lowry or Keldol, has any idea of the actual amount of protein. Enough on methods, results, and discussion. You report the bulk of your results once in a table, a figure, or in the text. You discuss only results that are statistically significant. A lack of difference, again, not absolute. I admit there are times when you want to say something about non-significant, but that is not going to be the guts of your paper. And think about scientific figures of significant, what's called significant figures. Remember that your standard deviation and your 95% confidence level is plus or minus 3 standard deviations. So look at that standard deviation and think about that number plus or minus 3 times that standard deviation. And then you'll see why significant figures, generally for biological materials, is going to be 3 significant figures. In many cases, it's only 2. If you have a high standard deviation, you may not be able to justify 3 significant figures. And I've written out a set of 3 numbers, all of which are 3 significant figures. Basically, if you use scientific notation, and in that case, I've used 4 significant figures. The 10 to the nth doesn't count. What counts is those 4 numbers. That's 4 significant figures. And biological materials with most test procedures do not justify 4 significant figures. And the standard deviation should probably also be adjusted. There are texts that go through this with rigorous rules. I tend to kind of keep it a little simpler, but again, I really check papers for significant figures when I'm reviewing, certainly when I was the editor. And remember, all of our data is really we're interested with standard deviation, the precision of the number. What we never really fully know is the accuracy, unless we have other pieces of information. So again, you can do Keldol to a reasonable amount of precision if you're very good at it and it's repeatable, and your sample is actually allowing you to do that. We did some work with Keldol at one point early in my career. One of the students in the lab, I was still a student at the time, prepared samples, sent them to testing labs, and they came back from all the testing labs with 5 or 6 significant figures. And the same sample, the numbers range from 9% protein to 15% protein. It's not easy to get accuracy. They were all precise, but because the raw material sample could not be made uniform, the sample sent to each of these companies was actually different. Reporting of the results should be in a logical sequence. It does not and should not be necessarily the order in which you did the experiment. Now, sometimes you specifically did experiment A and it led to experiment B, which led to experiment C. Then the story goes that way. But if you're using multiple techniques, you may have done some of the spectral techniques with different equipment and different aspects of the spectral at different times. But again, you may have a story to tell about the spectral properties of your material, then keep them all together. You're not tied to that original order. The odd thing is you're telling it in the third person. You're telling it as a storyteller looking in on this. And that's to give you the right to kind of look more carefully as an outsider at what you did. And again, English is a terrible language, but we have tenses. Most of the work was done. Most of the papers you're citing were done. They're history. They're past tense. The stuff in the paper is there in front of the reader. So that's in the present tense. Figure 1 shows. So-and-so's results showed. Or in the passive tense, it was shown that something occurred. It's tricky. I know English is terrible. That's why it's often useful to get help. Then there is the discussion, usually best to do for each experiment. That is, if you're doing spectral techniques and you're doing UV visible and you're doing Raman and you're doing FTIR, do a discussion of that piece so that the reader understands what to take away from that, and then have the spectral discussion. And then at the end of the results and discussion, or in some journals as a separate, then you pull things together from the entire paper in that final discussion. And your conclusions then are what you have done that is new. Why is it important? What have you learned? What limitations are there on the conclusion? Suggestions for future work. The conclusions is a focused framing of, I've told you my whole story. This is what I want you to take away. It is more focused than the abstract, which is covering the whole paper, but it is giving people the really important scientific message. Then there's a whole bunch of boilerplate, conflict of interest statements. Take it seriously. I've seen one or two where there are conflicts, and it's fine. The authors were upfront about it. I was able to have whatever follow-up was necessary, and they were fine. And it made it a much more credible that they were honest about it. Acknowledgments. Somebody paid for it. Even if you did it at the university with no funding, thank the university for making your office and your paper and your being there possible. And who helped? That is not an author. A lot of people sometimes do very important things and are very helpful. They're not authors. They deserve to be acknowledged. And nowadays, there's been some interest in AI statements, so I've given you an artificial intelligence statement and again, it has to be correct. This is what we're using. I do not use AI actively, but it turns out that the famous Word, the Microsoft Word program is clearly using an AI grammar checker that's different from what it was 5 years ago. So I share that. References. References are hard. You have all kinds of programs that keep track of your references. They only are as good as the information you put in. And the format of a journal is nitpicking. It's very, very structured. You need to know what the format of the journal is. And that's why I also encourage you to be sure to pick the right journal. Because every time you change journals, you may have to change the formatting of your references, both in the text and in the reference list. And it's references in the reference list. And the order and the capitalization, the use of italics, the volume, the pages, are you writing out the journal name? Are you abbreviating? All of those details have to be consistent. Are you putting a space between people's initials? In other words, I'm J dot space M dot, or am I J M, no dots at all? Or am I J dot no space M dot? They're all different. And you need to be consistent both internally and consistent with the rules of the journal. Do not try to manipulate citations again to affect the impact factor or your citation factor, your personal or the journals. Be careful with italics. Most of the time, journals are italicized, genus and species are italicized, in vivo and vitro are italicized, particularly in the references also. Non-journal articles is what will drive you crazy. They need to be formatted as similarly as possible, dealing with page, publisher, location of publisher, title. Is it going to be a title in italics? Is it all caps for the title? Is it only the cap of the first word and proper nouns that require capitalization? It's a pain. It is really, really testing how well you can look at something carefully. Tables, are they needed? In the old days, they took typesetting. Nowadays, there's a little more leeway because they just take your table and print it. So you got to be a lot more careful too when setting it up. Can you just put it in text? I've seen a table that has the approximate composition of one item. That should go into the text. Are the footnotes clear? Are they complete? Have the standard deviation and statistical analysis been included? And make it look good. People, again, you're making an impression on people. And think about what goes in the heading and goes in the footnotes. Look at previous papers in the journal you're going to. Sometimes they have a kind of style, and the better you set it up to that style, the better it is. Figure legends, there is a thing such as a figure legend page. All the figure legends go on one page. That goes back to when things were typeset. If you're using a figure legend page, then each figure should have its number, but nothing else. Do not have two sets of figure legend, one under the article, under the figure, and one on the figure legend page, because if corrections are made, people get sloppy and miss one of them. Use black and white. Color costs extra in print. Now we're seeing more and more journals online where it doesn't matter. But pay attention. Are you paying extra for color? If you're looking at a picture, electron micrographs are common. Is this what you see all the time with everything in your sample set? Or is this a really odd thing that you've only seen once? Both of them are okay. Just tell the reader, we saw this once, but we believe it may be important. This is what we saw most of the time, or this is what we really saw all of the time. All of them are a judgment you make as a scientist, but let the reader know that what judgment you made. Again, figures are potentially expensive in print. Are they necessary? And again, figures are not an excuse in cases like bar graphs or things of that sort to use a figure to get away with not having to do the statistics. You still need to do them. Again, if you're doing an electron micrograph or a visual of your material, that obviously doesn't, but think about that. Few other things as we quickly, hopefully, lined up. Seek help for English. Define your abbreviations first. Time they're used. Things like chemicals and amino acid name. H2O does not need to be defined. Calcium chloride does not need to be defined. Gly, trip, Lu, etc. The amino acid three-letter names do not have to be defined. But if you're going to use them, then you have to use them. Be consistent. I have seen papers submitted with using a big L and a little l for leaders in the same sentence. You know that that person didn't pay attention to their paper. You can quote things. That is not plagiarism. If you quote something, you can use it as is. You don't have to play games. You cite the author. It is not plagiarism. It is a lot better than trying to take and fudge the wording of somebody so you don't have to cite that you took it from someplace else, because it may well be picked up as plagiarism. And that's my final reminder, I believe. Plagiarism, data manipulation, and things like that. Don't even think of it. You can ruin your entire career by making such a mistake. Science counts on our integrity. Ethics and integrity count. So be careful. Oh, I got a few others. Use continuous numbering. Focus the title. Avoid filler words. Again, do not constantly say, and so had a study. Give the author showed and get right into it, or give the information and give the reference. You don't have to use a lot of extra words. Again, once you've established the samples, maybe in the results, you mention the samples, but if the samples are obvious, you don't have to keep saying, we have, 5 samples and the third sample. People know what you're talking about if it's clear without having to constantly repeat. And remember that modesty thing. Let the reader decide that you're the greatest thing and that they're going to put you in for a Nobel Prize. You don't have to tell them to do it. Thank you for listening. I hope this was helpful. I hope we have a little time for some questions and answers. Let me get back to, hold on. And Claire, I guess, has a question.
Yeah, Joe, Dr. Regenstein, great presentation. I really enjoyed it, very thorough. I didn't want to interrupt you when you were talking about authorship, but I was wondering if you could comment on perhaps how considerations for authorship and around your discussion around corresponding authors might be changing with new kind of journal requirements to actually put contributions of each author to the paper, at the end, and
Yeah,
how, yeah, just
that I
maybe.
think helps a little, but that's a place where I'm still convinced people are not. I don't want to say people are lying, but I think they may be stretching the truth at times. And
Okay.
I end up, I have, I am on a number of papers post retirement. I am on a ton of papers. I have never asked an author to put me on their paper because of the work I've done on their paper. In many countries, the culture is such. I tried fighting early on and learned that it just upsets people if I don't allow it. So I'm often put down as help with writing and reviewing. Um, do I deserve to be an author? I make I massacre manuscripts, so I don't know if that counts. I have stopped fighting it, I admit. I'm not always convinced that I should be, but I've read every word at least once and probably read it and read it closer than the people who actually wrote it. It's a very awkward situation, and there really is no absolute truth. Um, in certain countries, it's absolutely necessary that the director of the laboratory be included. Sometimes, and occasionally I've seen it honest, the contribution was getting the funding. And
Right.
that generally is their role.
Mm-hmm.
And but getting the funding, I mean, some of these big labs, you know darn well that the senior person is not carefully reading every paper coming out of his or her lab. What do you do if I don't have an answer, which is why I kind of send people to the NSF document. It doesn't have complete answers. It at least tries to set some standards, but it's set to what would be the American scientific standard. And in some countries, what they're saying is just culturally not going to fly. And I've just come to accept it. How can you have three corresponding authors? If the role of a corresponding author is to handle the correspondence, how can you have three corresponding authors? I have come to accept that because it is a requirement for the lab to get credit for the work that the senior person be a corresponding author. It's very frustrating. It's a very unsatisfactory piece of manuscript work.
Sure.
Is the issue of who is an author.
I have one other question if no one else has a question.
I haven't seen another hand go up.
Just curious, didn't hear thoughts on supplemental materials and, what, if you have thoughts on, the value of putting more information in supplemental materials, maybe some journals, it's more common in others, or some fields in my area of, developing simulation models, there's often a lot of methods in detail or supplemental code or things like in a supplemental file apart from methods. I'm just curious if that's useful to any students on the call.
Again, it depends on the work, it depends on the journal. I'm all in favor. If there is material that can be deposited in such a way that future scientists or people reading the paper can have more information and can play with the data in new ways. In science, we should be sharing as much as possible. And to the extent that supplemental data prevents padding papers with useless information from 90% of the readers, I believe it's a good thing, in that if you're telling your story well, all your readers who take the paper seriously will get the main pieces. A few of them will then want to see the detailed data to either manipulate it, to check that it was done, to evaluate it, etc. And that's a good way to do it, rather than pad, particularly print, which is getting more and more expensive. And even, again, to read many papers, if you're doing a review paper, all that supplemental material is probably not helpful, and therefore it probably is a good place for it. We're also seeing the use at Cornell. We now have, I had a student, I was a minor member from a totally other field, and she has a great deal of data that she's putting up on the Cornell depository. And when she's writing papers, she's referencing the depository rather than supplement, because she's going to have a number of papers with different framings. And that way, all the data is in one place, as opposed to still being in 5 different journals. So it's an evolving field. It's a good thing. I think it's appropriate for science. I'm not sure what is actually the best way to do it. And I think that's going to continue to change as the technology changes.
Great. Thank you very much. And thank you, Dr. Regenstein, for your presentation today. I mean, I think you covered a lot of great material, and there could be a lot more questions and comments people have for discussion from this. So if people have anything, you have your contact information on the screen. Is it okay if they reach out to you for further information?
I am happy to be in correspondence with anyone who wants to. Anybody who wants the slides set separately, I'm happy to do it. I give it as a PowerPoint. If somebody wants to use it, I hope they give me credit for it. But the important thing to me about information is to get it out there. Not, it isn't about my ownership. It's getting it so it's useful for others. So anybody who wants the slide set is welcome to it. And I'm happy to discuss any of these things. And I mentioned the guidance document, which is 20 pages. It's a lot of material, but it is useful for people in preparing. And again, happy to share that.
Okay, great. Thank you very much.
My
Just in
pleasure.
preparation for closing today's session, I want to remind everyone that our next webinar for Phi Tau Sigma Fall Webinar Series is December 6th, where we'll be covering abstract preparation and best practices for presentations from Dr. Moore, Dr. Otezi, Christina Ellingham, and Dr. Zollner. So if you're looking to register for this, please go to the Phi Tau Sigma website or look in the Phi Tau Sigma newsletter for the registration information. Thank you all and have a great day.
Bye now.